Hello everyone, welcome to the meiosis video. In this video we're going to talk about the process of meiosis and the processes that are involved in contributing to genetic variation. If you take a look at this picture right here, this is showing a sperm that's actively fertilizing an egg. And in order for this fertilized egg to become viable, it had to have obtained half of the genetic information from the sperm and the other half of its genetic information from the egg. And so meiosis is the process of generating these sex cells, so these particular gametes. And these are just some goofy but fun cartoons depicting sperm and the process of fertilization. Okay, before we discuss meiosis though, I want to clarify some vocabulary. This is an image of a karyotype where the colors are representing the individual pairs of chromosomes, and this would be a karyotype of ourselves. And if you remember, we have 23 pairs of chromosomes, which would also mean we have 46 chromosomes. And the new vocabulary word that we're adding is the term diploid. So we don't just have one copy of a gene, we have two copies of a gene. And so a diploid is essentially a cell that has two copies of every gene. And it's the total amount of chromosomes that will exist in a particular organism. It's also represented by this formula 2n, where n equals the haploid number, or half of the genetic information. And R2n equals 46, where our n equals 23. So the next thing I'd like to discuss is the human life cycle. So meiosis is really important because it contributes to genetic variation. And this is why you don't look just like your brother or your sister, and you don't look identical to your mother or your father. And that's a good thing, because we want our offspring to be a little bit different. Because if we were all the same, then if we had maybe something bad in our genes, like a disease, or maybe we weren't well adapted to the environment, then that would also get passed on to our offspring. So one of the greatest things of meiosis is that it contributes to that genetic variation. This is depicting the human life cycle, where you can see an organism here who has reached puberty and reproductive maturity. Through the process of meiosis makes eggs. And another organism, well, of a different sex, will undergo meiosis to produce sperm. So you can imagine this was your mother and this was your father, and when your father reached puberty, he was generating sperm. And for all the females out there, we were born with all of the eggs that we will ever have. And we don't manufacture new eggs. Instead, once we've reached sexual maturity or puberty, we will release our eggs, or our eggs will mature and they will be released by our ovaries. And so these are also called gametes, which are sex cells, will fertilize and produce a zygote. And a zygote is a fertilized egg. And through the process of mitosis, this zygote will grow and become an organism who will eventually reach sexual maturity at one stage. And this whole cycle repeats itself. I would like next to talk about mitosis, just so that we can compare mitosis with meiosis, because it's important to compare these two at the same time so that you don't confuse mitosis with meiosis. Let's imagine an organism that has a 2n equal to 4, which means it has a total of 4 chromosomes. And this organism has inherited 2 chromosomes from mother, which is represented in red, and 2 chromosomes from dad, which is represented in green. So the first thing that has to happen in this organism is we need to see DNA duplication, which takes place in S phase, if you remember. So now all of the chromosomes are duplicated so that this organism can set up a scenario where it can pull those chromosomes apart and make 2 identical daughter cells. And so these chromosomes will then align at the center of the cell, and that by the process of microtubules binding to the centromere, which is that center of the chromosomes, the sister chromatids essentially, and pull those sister chromatids apart, so that what happens is the creation of two daughter cells that are now identical to the original parent cell. So that's mitosis. That was just review. Now let's look at this organism undergoing meiosis. Okay, so here we have the chromosomes lining at the center of the cell. Notice that instead of chromosomes lining up like essentially on top of each other, okay, like how mitosis is, the homologous chromosomes actually attach to each other. Now, when homologous chromosomes attach to each other like this is called a tetrad. So a tetrad are duplicated homologous chromosomes that have now formed a bond with each other. This bond that is formed is almost like a protein network or web-like network that bonds these chromosomes together, and this is called synapsis. So synapsis is the actual process of these homologous chromosomes binding to each other, and these tetrads are bounded so tightly that they begin to exchange genetic information. And I'm going to explain that after I explain this slide. But we see the tetrads actually forming aligning with each other. So the tetrads line up with each other. So they're not the individual chromosomes that line up. The homologous duplicated chromosomes will line up on top of each other. So in this scenario, the cells are not finished dividing. And so the other thing I want you to know about meiosis is that it undergoes 2 rounds of cell division. The first round of cell division is called meiosis 1, and the second round, conveniently, of cell division is called meiosis 2. The great thing about the substages of meiosis is that it's identical to that of mitosis, where we have prophase, metaphase, anaphase, telophase all happening. We just define the substages by the round of cell division that the cells are in. So if the cells are in the first round of cell division of meiosis, which would be meiosis 1, then the substages are defined as prophase 1, okay, metaphase 1, anaphase 1, etc. And that's the same thing for the second round. So meiosis 2, it's called prophase 2, metaphase 2, etc. And so these 2 cells are now going to be represented here, okay? And the chromosomes, how they line up is essentially like how the chromosomes line up in mitosis. So they are on top of each other like this. And through microtubules binding to that sister chromatid, okay, we're going to see chromosomes getting pulled apart so that this one cell now becomes 2 cells. And the same thing happens with this cell, where this cell will generate another 2 cells. Now these 4 cells, okay, are distinctly they're very different from the original parent cell. So what makes meiosis so different is that it first undergoes 2 rounds of cell division. The second thing that makes it so different is that in the first round of cell division, we see synapsis taking place. So those homologous chromosomes are forming bonds with each other. Another major thing that happens in meiosis that's different than in mitosis is that 4 cells are generated, and those 4 cells are haploid cells. So they have half of the genetic information than the original parent cell. And so meiosis is the process by making these haploid cells. So we want to make cells that have essentially just N. And this organism will have an N of 2 because it had a 2N of 4. And that's why there are 4 cells here with only 2 chromosomes. So now let's take mitosis out of the picture, because we're not discussing mitosis. We're discussing meiosis today. And I want to focus on this cell that's right here. Now, I mentioned before that in the first round of cell division of meiosis, the way it's categorized, the subphases, are identical to that of mitosis. It's just that we have a 1 at the end of it. And this is what happens in what's called prophase 1. So in prophase 1, we see those tetrads forming, and we see these homologous duplicated chromosomes forming a bond. And that's the synapsis. The next thing I want to discuss is how these chromosomes can assort. They don't have to assort this way. So it doesn't have to be that all mom's chromosomes go in one in one cell and all dads goes in the other two cells. That's not how it usually works. And this can be completely random how these chromosomes align. So you can sometimes get something that looks like this, where you have dad's long chromosome here and mom's short chromosome here, so that these four cells will have a mixture of mom and dad's chromosomes. So that can happen too, and actually it does happen. This is called the law of independent assortment. So independent assortment is the idea that the chromosomes will orient themselves independent of each other, so that you could get dads, like this one is not dependent on these chromosomes here. You might get dads here and then moms here. So that's independent assortment, and that helps with genetic variation. The other thing that you would see that can happen in prophase 1, and it happens quite often, is the exchange of genetic information between or across these homologous chromosomes. So it can look something like this. So just notice that we see some of mom's chromosome, like the tip of mom's has a little bit of dad's, and some of dad's chromosome has mom's. And what we see over here is a larger amount of genes getting exchanged between mom and dad. And this is called crossing over. The process of crossing over is when the tetrads form those bonds that are so tight with each other that now they can exchange information. These chromosomes are homologous, which means that homo, which means the same, and logos, which means relationship. It means that those genes have such similar relationship, like they're similar in the structure and the function and the coding of that information, that they can swap places with each other. So this happens quite frequently. And this will happen in the early stages of the first round of cell division of meiosis. And so this cell right here that now has a mixture of both mom and dad's DNA and almost all of the chromosomes will generate 4 cells that are very different from each other. So you get a little bit mixture of mom and dad's information everywhere. And so this is completely random as well, and this helps contribute to variation. So let's take a look at those variations and pull them all together and discuss those 3 major processes once again. The processes that contribute to variation, that very first one is independent assortment. That's the idea that these chromosomes will assort independently of each other, which means chromosome 1, which is the long chromosome in this case, does not depend on chromosome 2. It doesn't matter if you have moms all on this side or dads all on this side. It's completely independent. And you might think that this isn't a big deal and doesn't really contribute to a lot of variation, but it does once you start adding more chromosomes into the picture. So we have 46 chromosomes, which means we have 23 pairs. If you take 2 to the 23, that's how many combinations we can make as a result of these chromosome pairs not being dependent upon each other. Another important process for contributing to variation is crossing over. These homologous chromosomes will form synapses, which bonds these homologous chromosomes together. And they bond so tightly, and they actually they are so tightly wound, they sometimes can wind around each other really tightly, and when they do so, they can exchange genetic information, such that now you have the case where a little bit of mom's chromosome over here is on dad's, and a little bit of dad's genetic information is now on mom's chromosome here. And so that helps contribute to variation. Another important feature, and this is the last one, is that fertilization is random. The sperm that fertilized the egg that made you who you are, it was completely random. And it definitely was the luckiest sperm out of the bunch. All right, so that's meiosis, and I hope that was helpful. And as always, keep studying.